Hello and welcome everyone. Today we shall discuss a very interesting topic, that is SD, LDL and modified LDL. As you can see, these different particles here, these are known as the different types of LDLs. So let us begin with low density lipoprotein. Low density lipoprotein particles are derived from the metabolism of VLDL, that is very low density lipoprotein, and they are enriched in cholesterol. So LDL or low density lipoproteins are enriched in cholesterol. The LDL particle is made up of phospholipids and cholesterol that form the hydrophilic surface. So this is the phospholipid and this is the cholesterol which forms the hydrophilic surface. The cholesterol ester and the triacylglycerol form the core, and this core is hydrophobic. There is one protein, that is the apoprotein B100. This apoprotein B100 is essential for uptake of LDL particles by receptor mediated endocytosis for further metabolism. So this is the general structure of an LDL particle. Now, what are the functions of LDL? LDL is a bad cholesterol and it transports cholesterol from liver to peripheral tissues. The transported cholesterol has various fates. So, this cholesterol which the LDL transports is used for synthesis of steroids. They may be incorporated into membranes and they may be esterified to monounsaturated fatty acids and stored. The LDL consists of several subclasses of particles with different sizes and densities, including the large buoyant LDL, intermediate LDL, and the small dense, that is SD LDLs. So these are different subclasses of the LDL which are formed during the LDL metabolism. So overall LDL is known as bad cholesterol. This is a misnomer because it transports cholesterol, and it is this cholesterol which may get accumulated into the arteries causing atherosclerosis. Now let us see the small LDL, which is a subfraction of LDL. So this small LDL is also abbreviated as SDLDL. This SD should be written in small letters. It is highly atherogenic. SDLDL particles enter the arterial wall easily compared to the large native LDL particles. They bind more strongly than the large native LDL particles to the intra-arterial proteoglycans. The small LDL particles are also more easily oxidized. They have a long half-life. The modified forms of SDLDL are pro-inflammatory and are prone to aggregation and formation of complexes that further increase their atherogenicity. So, SDLDL or the small dense LDL are far more atherogenic than the native or normal LDL. And the reasons are that they easily cross the arterial wall, they can bind very strongly to the proteoglycans, and they are more easily oxidized, and they have a long half-life due to low affinity to the receptor. Now we'll see the next combination, that is the modified LDL. The modified LDLs, actually these LDL particles which are originally produced from VLDL are known as native LDL. These native LDL particles undergo several modifications that change their biological properties. They are known as modified LDLs. The multiple modifications of LDL particles have been detected in human plasma. The modification of LDL is a critical event in. Now Now these are different species of modified LDL which are produced from native LDL. When a native LDL undergoes certain reactions in the body, that LDL is modified and it is known as a modified LDL. So let us look at those different modified LDLs. So this is the oxidized LDL, which is the first species. It is oxidized by reactive oxygen species, reactive nitrogen species, cupric and ferric ions. Any one of these agents can oxidize the native LDL to form oxidized LDL. The second group is the enzyme modified non-oxidized LDL. So the native LDL is modified by phospholipase A2 and or cathepsin to form enzyme modified non-oxidized LDL. The third kind is the desilyated LDL, and the desilyated LDL affects endothelial permeability in atherosclerosis. So what is desilation? Desilation is nothing but removal of sialic acid. And once the sialic acid is removed, this modified LDL becomes highly atherogenic. The fourth kind is known as the glycated LDL. So when the native LDL reacts with the excessive glucose molecule, it forms glycated LDL. The glucose is derived from hyperglycemia or diabetes mellitus, and it attaches with the LDL to form glycated LDL. And the fifth type shown in this picture is known as the carbamylated LDL. This carbamylated LDL is a modification of native LDL and it is formed from the thiocyanate from smoking or nicotine smoke. So this also carbamylates and modifies the native LDL. So these are the different 5 species of modified LDL. There may be many more. So this is the endothelial layer of a blood vessel. This is the native LDL which is produced from VLDL. This native LDL is carrying it's carrying cholesterol. Now, when this native LDL is subjected to various chemical reactions such as oxidation, glycation, enzyme modification, desilylation and carbamylation, which we saw in the previous slide, they are modified to modified LDL. And this transformation of the native LDL to modified LDL attracts autoimmune response. So the immunoglobulins come and attach to this modified LDL, and this makes the modified LDL a large immune complex, and this large immune complex has high heterogeneity because it can easily cross the endothelial layer, and this is how the process of heterogeneity begins. So the modified LDLs are far more heterogenic compared to the native. So this is the native LDL, and now we will be summarizing the entire process after knowing what are small dense lipoproteins and modified LDLs. So this is the native LDL. It is produced by the VLDL. Now this LDL will undergo modifications by various reactions which we saw in the previous slides. Once this LDL is modified, it will form aggregates, and these are known as modified LDL self-associations. And these modified LDLs which form aggregates are taken up by the macrophages by phagocytosis. There is release of pro-inflammatory cytokine secretion, and as these modified LDLs enter into the phagocytes and their number increases, they expand in number, there is foam-like appearance in the phagocytes, and so they are known as the foam cells. The native LDL do not form foam cells. The modified and small dense LDL form foam cells. Foam cells in the beginning are not dangerous, but as their size increases and their number increases, their excessive accumulation of foam cells leads to necrosis within the atherosclerotic plaque. They cause the hardening of the arteries. So the what is a plaque? A plaque is made up of deposits of fatty substances, cholesterol, cellular waste products, calcium and fibrin. So we will see another diagram. Now, this is a normal artery where blood is flowing. There is no atherogenesis. And here now, due to the modified LDL, there is built up of plaque and foam cells. And this is a stable plaque. But as the uh, foam cells increase and various inflammatory reactions proceed, the plaque becomes unstable and it ruptures. These are the events of atherosclerosis. So now you know that the modified LDL and the small dense LDL are far more atherogenic than the native LDL. So because of their atherogenicity, high atherogenicity, they are attracting the attention of um, all physicians as they are very important atherogenic factors and they are independent risk factors and independent markers of atherosclerosis and future events of AMI. So these are my references. Thank you. Please like, comment, share and subscribe to my channel. Thank you for watching.